Okay, hope you can see the screen clearly.
Yes, we do. Yes, we do.
All right. So thanks. I am Onur from the Max Planck Institute. Well, at least I started there, and now we have moved on. So yeah, I will be talking about the diversity of pathogens and how this diversity in turn shapes and affects our immune diversity. Now, humans, just like any other organism, lives under the constant assault of many diverse different pathogens. And of course, our main defense mechanism is our immune system. And my research is more focused on the adaptive side of this immunity, and more specifically the MHC genes. The MHC genes, which is short for the major compatibility complex, encode for the molecules that present antigens on the cell surface. And if these are recognized as foreign, then the adaptive immune response is initiated. In humans, these genes are also named as human leukocyte antigens. Throughout my talk, whenever I say MHC or HLA, I will be referring to the same cell surface molecules. Now, there are interesting features of MHC genes, one of them being their exceptionally high diversity. We can find hundreds, even thousands of different alleles for almost every loci in humans. And not only the number of genes are high, but also the number of differences between these alleles, not only the number of alleles is high, but also the differences between these alleles is so high. And most of these differences are concentrated on the region called the peptide binding region. And the residues that encodes for this peptide binding region has much more non-synonymous mutations than these synonymous mutations, indicating the act of selection on these residues. And compare the frequency spectrum of SNPs that are within the HLA region to those that are outside the HLA, which are the neutral SNPs. We observe an excess of intermediate frequency variance within the HLA SNPs, for the HLA SNPs, but not for the neutral SNPs. So all these and several other observations to the hypothesis that balancing selection of MHC in the adaptive immunity, pathogen-mediated balancing selection hypothesis has been framed. The main assumption of the pathogen-mediated balancing selection hypothesis is that different MHC variants binds different antigens. We know that this is true from many different experimental work, but we need some more insight about how these antigens are actually distributed or shared between different pathogens. So we may consider different scenarios. For example, if the sharing of antigens between different pathogens is high, then a single MHC allele that can bind to these shared antigens might be enough to mount an effective immune response against all these pathogens. But if the sharing is too low, then we will require distinct MHC molecule, potentially recognizing different pathogens. So our first aim was to characterize this antigenic diversity of pathogens. And we have, to do that, we have selected 36 representative human pathogens, most of which were already compiled by Federica and Reem from our lab. And I have simply downloaded the proteomes, and we have obtained with a sliding window approach all possible 9 mers of these pathogens, which represents potential antigens that our MHC molecules can bind. And in total, we have obtained more than 51.8 million 9 mers, and we have analyzed the sharing of these 9 mers among the pathogens. So what we found was that almost 99% of these 9 mers were unique to the pathogen from which they originate. And only slightly higher than 1% of these 9 mers were shared by 2 or more pathogens. And we have also analyzed the peptide sharing among pairs of pathogens to see how much of their complete peptidomes are shared within pairs. And we have found that most pairs only share really small number of their peptides with the other pathogens. And this sharing was this pairwise peptide sharing was negatively correlated as expected with the evolutionary distance, indicating the role of homology, the sequence homology that creates this, that leads to this peptide sharing. So we have, in a sense, shown that the antigenic diversity of human pathogens is so large that almost every pathogen potentially challenges the immune system in a new way. Now, the next step is to find out how this antigenic diversity shapes our immune genes, our immune diversity. So to do that, we have employed an in silico approach. We used an established binding prediction algorithm, namely the NetMHC-PAN. This is a machine learning algorithm that is trained on experimental binding data, and it allows prediction of binding affinity between any MHC molecule and any peptide that we provide. So we have collected 321 common HLA alleles by using the common and well-documented alleles dataset, and simply calculated the binding affinity between each allele and to the complete proteome dataset, the peptidome data of our pathogens. So this is the distribution of the number of bound peptides for the HLA-EB alleles. So here on this axis, we can see the names of different HLA-EB alleles, and here is the number of bound peptides by each allele. Of course, we can the variation is really interesting, and we have like alleles on one hand that binds really high number of peptides, which we call the promiscuous alleles, while on the other hand we have these fastidious alleles which binds small number of peptides. And the question that comes into mind is how these fastidious alleles can be maintained in the populations while their promiscuous counterparts binds much higher number of peptides and potentially provides resistance against like more diverse pathogens. One hypothesis that can answer this question is provided by Jim Kaufman, and he will have a talk specifically on this topic tomorrow, I think. So I apologize that I have to give a bit of a spoiler here, but otherwise my results would not make much sense. So he suggests that these promiscuous alleles might act as generalists that provide resistance to common pathogens, while fastidious alleles might be specialists that become important in the events of outbreaks or in the event of consistent pathogen pressure. So we wanted to test that in a quantitative way. So we have analyzed the proportion of bound peptides by each allele from each pathogen. And we have normalized these values so that different alleles will be comparable to each other. And here is the like a summary, a heat map that kind of summarizes our results. Now, again, in this axis, we can see the alleles, and here is the 36 different pathogens in our data set. The red color indicates that the specific allele binds like high number of peptides from the corresponding pathogen, while the blue color means the allele binds small number of peptides from the corresponding pathogen. Now, overall pattern is that we can see mostly the light colors, which indicates most of the alleles binds reasonable amount of peptides from most of the pathogens, but we can still observe some extreme values. To analyze these extreme values, we have come up with a specialization score. So let me show you with an example what this specialization score or specialization value is. So for this allele, we can get the difference between the median and the maximum binding values as a specialization value, which shows how much difference is there between the most like highly covered peptide and the allele's overall binding capacity. So we have calculated this specialization value for each allele in our data set, and we have shown that this specialization value is actually negatively correlated with the promiscuity, meaning that the alleles with the high specialization value tend to be more among the fastidious ones. So to conclude, we have shown that the overwhelming majority of these 9-mer peptides were unique to the pathogen from which they originate, and this leads to the conclusion that most pathogens challenges our immune system in a new way. And we have we did not observe, and we of course were not expecting to observe one HLA, one pathogen relationship, but we can still see some indications of specialization for some HLA alleles, specifically for the fastidious ones. And such specialization might indeed be one of the reasons how these fastidious alleles are maintained in populations. So with that, I would like to thank you for listening to the members, like current and the past members of the evolutionary immunogenomics group, and to Jim Kaufman for really insightful discussions. Thanks.